So coming to the topic, as I have been introduced by Dr. Saab, so the conclusion or the point is the study, it has a therapeutic potential to prevent psychoneurological diseases. And I would like to introduce a little bit of study. This is the topic of my study, which is toxoplasma gondii infection. It alters the metabolic profile of maternal-fetal interface. And as we know that toxoplasma gondii, it's a parasitic disease. And our main objective was to see that how it is transferred from mother to fetus during different phases of pregnancy. So our prime objective was this one, but during the course of study, this whole pathway was directed us to a new direction, to entirely a new direction of metabolomics or microbial metabolomics, and then psychoneurological diseases. So we will see that by one. So first of all, I would like to just a general mind map of that what we did in this study. It was an LCMS-based study in which P-Helix and C18 columns, they were used with the two-step extraction protocol was adopted for the extraction of different metabolites. And both global and targeted metabolomic studies or profiling were carried out during this study. And of course, as we all know that in metabolomics, one of the most important feature is to confirm the identification of metabolites. It should not be like putative, so it should be annotated. So I did that with the help of like MS1, MS2, and then a little bit of fragmentation data from Glasgow polyomics facility. And then, of course, the ultimate biological interpretation. Okay, so this slide, it shows the workflow. It was carried out in pregnant BALB C mice. And this study, it was carried out in University of Porto, which is in Portugal. So all the pregnancy maintenance, their housing, and everything was maintained in University of Porto. And then they were given toxo infection with ME49 strain. And after the infection, confirmation of infection, the tissues, they were collected or extracted from the mice. And those tissues were fetus, placenta, decidua, and serum. So this was initially, but later on in the later stages during the study, we also collected or worked on different types of brains as well due to the link with the psychoneurological disease. I will explain later on that why we did that. Anyway, so after this, the next step was these samples, they were shipped to the UK in University of Strathclyde, where HPLC and metabolite identification and further data analysis and everything was carried out here in the UK. So, first of all, I would like to show some of the difficulties which we encountered during this study, as we can see from the PCA plot here, that this one was a detection of an outlier in the data, and the corresponding loading plot, which is this one, green one. So it was showing unequal distribution or unfair distribution of different variables. So it was confirmed that for the whether it is an outlier or not. So it was confirmed with different methods, which is one of them is TIC value, total ionic current, both in positive and negative modes. So after the confirmation of, as we can see that this one, GW25, this peak of this total ionic current, it corresponds to the outlier in the PCA plot. So after the confirmation of this outlier, then again it was removed and then the plot was drawn. And as we can see, it's a very beautiful loading plot was carried out. So which shows the equal distribution in two groups, whether infected and control groups, and variables are uniquely or equally, they are distributed among in this plot. So similarly, in the fetus, there is one more aspect. As we can see that in many studies, there are OPLS DA plots. They are being shown, but as we can see, even PCA, it's not like ideal plot, whatever we should be expecting. There are some of the fetal units, they are not infected and they are behaving just like controls. But still, we can see that in OPLS DA plot, it shows exactly and very unique and very very beautiful distribution. So such type of graphing or such type of data representation in different literature, that is not fair, because of the reason that, as we all know, that OPLS-DA is a supervised pattern which exaggerates the distribution. Anyway, so the point of this slide is that if PCA plot, it is fairly distributed, then we should go for OPLS-DA. Otherwise, it should not be carried out. So when this PCA was post-mortemed, and it contains 3 different samples infected, which one, like they were named M4, mouse 4, mouse 5, and mouse 8. So from these 3D plots, we can see that this M8, it was a problematic sample, which were disturbing this PCA. So when this M8, it was removed, and then we got this PCA plot, which is a fair distribution of the samples or variables or whatever we should be expecting from the PCA. So this was the just due to the presence of M8, because out of many fetal units from the mouse, so this unit M8, mouse 8, or fetal unit 8, it was not carried out, it was not carrying any infection, or maybe the threshold of the infection was not up to the mark to classify into the diseased one. So similarly, the next one is placenta. So placental multivariate analysis, it was okay. And these 2 plots, these are from 2 different columns. First the A and B, or PCA and OPLS-DA. On the upper side, they are from the P helix column, and the bottom one, they are from the C18 columns. And after that, there was volcano plots were drawn, and these all were different metabolites shown in the pink dots, and they were extracted, like they were significantly changing. And then a list of such type, they were made in the Excel sheet. Although this is just for showing here, like this is very short list, but we had many Excel sheets, Excel pages of those metabolites which were significantly changing. And then we manually curated something, and we were only focusing on our desired one. But this study, it was an initial experiment, and we were doing untargeted metabolomics. So after this, then there were peak selection criteria, that how we would make sure that the different metabolites identified, whether they are true identification or not. And for that purpose, first of all, we need to identify the different types of peaks. And as we can see in the upper row A, these are different types of peaks shown here, which were selected for further analysis, or these were candidate metabolites or candidate molecules which were selected in the study for further analyzing those. And in row B, these are 3 different types of examples I have shown here, which types of peaks were not selected. Of course, this was no peak shape in zigzag or no obvious peak. And in the central one, as we can see that there is a high background, and the last one is it doesn't match with the retention time. So these are just only 3 examples, but there were many others, as I have mentioned here, confidence level of confidence, which were automated in the different softwares which I used for analysis. I used EDM software. So in that, confidence level is given to each metabolite from 1 to 10. So I selected only the metabolites which were having confidence level of 6 or more than 6. So any confidence level or any metabolites with the confidence level lower than 6, they were rejected or they were not included in the further study, because in the next step, we were going towards the targeted metabolomics. So after this selection, then there comes the confirmation of identifications that whatever metabolites we selected or we identified in our study, whether those are confirmed, whether those are putative identifications or like annotated identifications. So for that purpose, we did use BALB browser, which is a Tox ID software. So in that software, I used BALB browser, which gives like their retention time, their delta ppm, their MZ mass as compared to their standards run. Because as we know that for each of the metabolite detected in any metabolomic study, so it's not economical to run against E standards or it's costly. So few of the metabolites like maybe 156. So those standards were run against their like retention times and everything was noted and then compared with the putatively identified metabolites. So here I have shown some of the examples like this one. As we can see, this is standard sample as well as pooled serum sample. So then their retention time, MZ mass and delta ppm and their different ionic mode, whether it's from positive mode or negative mode, they were confirmed and they were identified. And then that boosted our confidence in the identification of those metabolites. So then we were making sure that, okay, this is the metabolites which we labeled it as whatever A, B, C, whatever it is. So similarly, another example of this metabolites identification, here we can see that this is from the negative ion mode. Again, the same standard, same procedure, same everything. So their retention time were noted, so they were compared. And in this, as we can see that their MZ mass, that's okay, that's fine. But in one MZ mass, as we can say that there are many hundreds or few of the metabolites, they can come under the same MZ mass. But then we can differentiate those metabolites through the comparison with the standards and to the RT, with the in respect of RT or retention time. So this was identified from the negative ionic mode. Similarly, next one is yes, there are many like maybe 1,200 of the metabolites were identified in untargeted metabolomics. Then those were selected for different step of the study, which were like targeted metabolomics. So there were like maybe 200 or more than 200 metabolites were selected. But one of the important feature is that some of the microbial metabolites, they were also detected in this study. As we can see that this is from the tryptophan metabolism, and tryptophan metabolism, it leads to the different types of indole production or indoxylsulfate. And this indole and indoxylsulfate, they were detected in our study. But one thing which was not sure initially, that it may be due to the hepatic metabolism, or there are some changes in hepatic metabolism, or it is due to the infection of toxoplasma gondii that it changes these microbial metabolomics and microbial community. But later on, it was confirmed in another study or further extension of this study that each time it was reproducible. It was like every time the same results were given by the toxoplasma infection, that these microbial metabolites, these are also changing. So it means this infection, it is also disturbing, or it is also interplaying with the microbial communities as well. And these are different: indole, indoxylsulfate, and indoxyl. So these were like three different indole derivatives or microbial metabolites. They were detected in each tissue, which were significantly changing in fetus, decidua, placenta, and next we will see that even in the serum. So this was also changed significantly. After this, microbial metabolites, then this is the slide from the targeted metabolomic studies. So first of all, we just need to focus on this fetus and serum, this Venn diagram of the fetus and serum, because then serum was also selected, and serum in comparison with the fetus. So these are different metabolic pathways like tryptophan degradation pathway, purine, arginine, and proline metabolism. So those were affected, and these are a list of different metabolites which were significantly whether downregulated or upregulated in the serum. But one thing which is quite obvious, that in serum there are more number of changes and higher number of changes as compared to fetus. So after this, then as we can see, this Venn diagram between fetus, serum, and brain. So this brain was selected because of the presence of tryptophan degradation pathway. Because tryptophan degradation pathway and ultimately producing kynurenine or disturbing the kynurenine level that is linked with the psychoneurological diseases. So then this brain was taken out, and this brain extracts, they were also included in this study. And these brains, these are taken from the fetuses, not from the fetuses, from the mice who were born to the infected mothers. And when they were adults, so then those brains were taken, and then those were compared. And exactly the same pattern of tryptophan degradation pathway or different disturbances in tryptophan degradation pathway were shown in the brain as well. So what are those pathways, or what are those metabolites, which is, as we can see, this is the tryptophan degradation pathway. And these are different tryptophan, alpha-formyl kynurenine, and kynurenine. Kynurenine is very much disturbed in brain, fetus, and serum. This is the graphical representation. So this was the main crux of this study, that this kynurenine production or increased kynurenine production, it starts early in the fetal life. And then it goes to the brain during the adult life. And as we all know that kynurenine is very much linked with the psychoneurological diseases or stimulation of psychoneurological diseases or during the adult life. So this was the crux of this study, that if we can stop this production during very early fetal life of the kynurenine production or high increase of kynurenine, then we can stop the development or like consequences which are like in schizophrenia or different psychoneurological diseases that can be prevented here if we can stop the production of kynurenine during early fetal life. Because the reason is, this brain is taken from, like I told you, this brain is taken from the fetus, from the different animals which were born to the infected mothers. But then we took brain from the adult animals which were born to the normal mothers, not infected mothers. In those brains, there were no disturbance or no alteration in any kind of tryptophan degradation pathway. There was, kynurenine was okay, that was normal. There was no any alteration in those. So which authenticates this fact that during early fetal life, this kynurenine production, it is linked with the brain abnormalities or for the development of psychoneurological diseases. So the take-home message from this study, which although in a literature there could be many other messages, there could be many other take-home messages or conclusions can be written down, but one of the main important thing is that if we can successful like to develop treatments to alleviate kynurenine production early in life could have an impact on prevention of psychoneurological diseases later in life. Yes, that's the main take-home message from this study. And that's all from this study. That's all from me. Next is your turn. And I'm very much open for your inputs, for your suggestions or anything, questions, please.